So here we are in Chapter 9, and we're going to start looking at skeletal muscle. Now, this chapter discusses all three muscle types: smooth muscle, cardiac muscle, and skeletal muscle. And I've got some pictures here for you to remind you of some of the characteristics of each of these kinds of muscle cells. But it will focus primarily on skeletal muscle. At the very end, we'll talk a little bit more about some of the characteristics of smooth and skeletal muscle. Smooth and cardiac muscle, but primarily this chapter is about skeletal muscle. So, muscle is each muscle that we have in the body that I'm going to be asking you to learn the names of is a distinct organ. It's its own individual organ, but they're composed of hundreds, maybe even thousands of muscle cells. And if you recall from some of our conversations back in Chapter 5 when we were talking tissues, we discuss or describe these skeletal muscle cells as muscle fibers because they're so long. So a skeletal muscle is composed of these muscle fibers, and here you can see in this picture we can see, here's a muscle fiber all looking way up close at one. We're looking inside the cell. These are individual myofibrils in the muscle fiber. But there's also connective tissue that's holding these muscle fibers in close contact with each other, proximity to each other, as well as electrically insulating the muscle fibers from each other. And then, of course, blood vessels and nerve fibers are found inside the muscle too, providing the energy required for contraction and the stimulus to contract. Most skeletal muscles are attached to a bone, and as they contract, they move these bones, but some of them are attached to tissues other than bones, sometimes to other muscles, or they're pulling against some other structure, and that's primarily in the face that we see these kinds of muscles. So some of the characteristics of skeletal muscle is that they are striated, and this is caused by the orderly arrangement of the thick and thin filaments that are found inside that muscle cell. So it gives the appearance, that striated appearance. And in a minute, toward the end of this lecture, we're going to discuss why they have that striated appearance. And they're voluntary. That means you can think them to move, so they respond to conscious thought and are under conscious control. And they tend to be very rapid contractors, but they use up their ATP stores, and so they tend to tire quickly. And that is in comparison to, say, something like smooth muscle, which is a slower contractor and doesn't tire very quickly. So when we look at the structure of a muscle, the first thing I want to point out are the connective tissues. So every muscle cell is surrounded by, and this is an image that's in your book, so I encourage you to find this image and turn to it and look at it. You'll have a little bit better view. But every muscle cell is surrounded by and insulated by a layer of connective tissue called the endomysium. And so here in this muscle cell, we got a muscle cell here. Here's another muscle cell. Here's another muscle cell. So this connective tissue that's all around them, there's a better view, is the endomysium. Perimysium is found surrounding a cluster of muscle fibers and creates what's called the a fascicle. Here's the term fascicle. So the perimysium surrounds a bundle of these muscle fibers and creates this clump of muscle fibers that are all bound together, first by their endomysium and then completely surrounded by the perimysium. And then last of all, the entire muscle is surrounded by a layer of connective tissue called epimysium. And this is a layer of dense, irregular, all of these are dense, irregular connective tissue. So they're providing good, strong support. The protein fibers are running in all directions so that they are really cinching down and very tough connective tissue wrappings that both insulate individual cells. So that's what the endomysium is doing, is providing this electrical insulation so that as we tell this muscle fiber to contract, this muscle fiber is not contracting unless we want it to. So that's the reason that we need to electrically insulate them. And as we come to understand better how we tell a muscle fiber to contract, then hopefully this will make more sense to you. So endomysium surrounds an individual muscle cell, perimysium surrounds a bundle of muscle cells that we refer to as a fascicle, and epimysium surrounds the entire muscle. And if you recall, epi means top, endo means inside, and peri means around. So hopefully that will help you to make sense of or to remember what their locations are. Then there are layers of fascia that are also associated with the muscle fiber. And so fascia, and that's how you say that, the SC makes kind of a sh sound, so it's fascia. The deep fascia is what we find here, separating and then binding together individual muscles. So if we think about this is the brachialis muscle that they're showing, it's overlain by the biceps brachii, and so this deep fascia is what binds together the biceps brachii to the brachialis muscle. And then there is also subcutaneous fascia or subserous fascia, and this is the fascia that kind of intermixes with the adipose tissue that's found insulating and protecting the tissue. So it binds the muscle fiber, or I'm sorry, not fiber, but the muscle to the that subcutaneous tissues that are associated with the skin, or if we're looking deeper in the body, that is associated with the serous membranes. All right, so fascia, connective tissue that's external to the muscle and that binds the muscle to other muscles or to the tissues above and below the muscle. So tendons are extensions of all of those connective tissues. I'm going to go back to look at this. So here we see a tendon. Tendons are this fascia, but they're also extensions of the epimysium, and then this endomysium, where if we look at, if we're looking at it from the side, so this is a fascicle, and here's another fascicle, and here's another fascicle. So where at the very end all of these fascicles kind of come together, and their perimysium kind of binds together with the overlying deep fascia and epimysium. And then inside of each fascicle, of course, there's all the connective tissues of that are surrounding the individual muscle cells. So all those connective tissues kind of run out at the end of the muscle and form the tendon. And so tendons are this super strong connective tissue where the collagenous fibers now become very organized, and they're all very regular. So the collagenous fibers in it are all running parallel to each other. And so if you remember looking at dense, regular connective tissue, that back when we were looking at the tissues, that's what this tendon material is. And it's this very strong, there's not a lot of elastic fibers in it, so it's not very stretchy connection between the muscle and the bone. And because the tendon is so integrated with the connective tissues of the muscle, it's very strongly connected into the muscle. And then where it meets the bone, the fibers, these collagenous fibers, reach into and interlace with the collagenous fibers that are found in the periosteum. And so it has a really super strong connection back to the bone as well. So tendons connect muscle to bone, but there are also these big broad sheets of deep fascia that form, that are here. There's one, we have these sheets on the lateral thighs, across the abdominal region. If we were to look at this dude from the backside, we'd see that there's a great big sheet of it that runs right down the center of the back, up around the shoulders. So this is all, I'm sorry, I got that the wrong place. Let's try that again. It's down the back, down the lower back and toward the hips. We have this big layer of deep fascia that is called an aponeurosis. If it's plural, it's aponeuroses. And they are also, they also attach the muscle, this time to another muscle. So aponeuroses are big, broad, fibrous sheets that attach muscle to muscle. Okay, so let's look now at the structure of a skeletal muscle fiber. Each one is a long cylindrical cell, and as you recall, it was multinucleate. And they're multinucleate because developmentally they form from the fusion of a number of much smaller cells. So we're looking here at an entire muscle cell. It's surrounded by the sarcolemma, which, by the way, is, well, we'll talk about this in a second, but apparently muscle anatomists didn't get the message about all the names of the structures of the cell. And so we're going to see that muscle cells have a lot of their own naming conventions. But what was I saying? So they're formed by the fusion of a whole bunch of cells, and they're going to be surrounded externally by that layer of endomysium that isolates them from any muscle cells that are hanging out nearby. So the nuclei tend to be ovoid, and so they've got lots of them, and then also lots and lots of, you can see these little yellow things, these are the mitochondria. So they need a lot of mitochondria in there to help make all the ATP that is required to run, to allow contraction to happen. Lots of other organelles too. We're going to look at that in the next slide. So some roots, anytime you see that root word sarco or the root word myo, know that they relate to muscle cells in some way, or to muscles in some way. Okay, so let's dive into some of this confusing terminology. And I hate it. I hate that this is, after I teach you all the names of the structures in a cell back in chapter 3, then we're going to switch over to this new naming conventions, but there it is. So hop on board and let's see if we can figure it out. So the outer plasma membrane, the cell membrane of a muscle cell, is referred to as the sarcolemma. so sarcolemma. And the function of the sarcolemma is not only to sort of contain and surround and do all of the transfer in and out of the cell that we associate with the sarcolemma, but it also has this function of receiving a nerve stimulus. So it receives a message from a neuron that it's time to contract, and then it spreads that message about contracting down the length of the muscle cell, and ensures that the entire muscle cell will do the contraction process. So sarcolemma is same as the cell membrane, and all the functions of the cell membrane plus it's involved in this communication function. Now, at spots along the length of the sarcolemma, the cell membrane dives deep into the cell. So here you can see a little opening. Oops, sorry. Here you can see this opening. Here's another opening here. Here's another opening. At these openings, the sarcolemma folds deeply into the muscle fiber, and it'll come and surround all of the myofibrils, they're called. So this structure here and this structure here is a myofibril. We'll see that definition soon. But the T-tubules, or the transverse tubules, are folds of the sarcolemma that wrap deep into the cell and surround, as a little tube, surround the individual myofibrils. And so I'm trying to see if we can see a T-tubule here along this length. But yeah, here's the opening. Here's where a T-tubule would come and surround that entire muscle fiber. Here's another one. So it would surround here. And so each of these T-tubules reaches into the muscle fiber and wraps around each individual myofibril. And these openings are concentrated at the junctions between the A bands and I bands, or at the points where A bands and I bands tend to be found together. And so we'll make some sense of that in just a minute. So T-tubules, just infoldings of the sarcolemma. Okay, so rather than cytoplasm, a muscle cell has sarcoplasm, and it's the cytosol plus all the organelles that are found inside the cell. And also included in the sarcoplasm are large amounts of glycogen stores. So we have these granules of glycogen, the storage forms of glucose, so that there's lots and lots of glucose available to all those ATP producing mitochondria. And there's also a pigment called myoglobin. That should sound to you a little like hemoglobin. So it's a reddish oxygen binding pigment that is found only in muscles, and it stores lots of oxygen there for ATP synthesis. So thanks to both the glycogen granules and the myoglobin, then we have all the stuff that we need to be ready to make lots and lots of ATP. So the myofibrils, so let's go back for just a second. So again, this is an individual myofibril. They run the length of the entire cell. So no matter how long the muscle cell is, then the myofibrils inside the muscle fibers are running that same length. So they are these thread-like protein strands, and they run the length of the fiber, and they run parallel to each other and to the length of the fiber. And there can be hundreds to thousands of these myofibrils, depending how large that individual muscle cell is. So the larger the muscle cell, or the reason a muscle cell grows in size, is because we add myofibrils to it as it's growing. So a person who works out a lot is going to have a lot of myofibrils inside each of their cells. And it's composed of two types of protein myofilaments. So myofibrils are built of myofilaments. There are the thick filaments. They're made primarily, or almost entirely, of the protein myosin. Come on. Stay small now. So, yes. So myosin. And here we can see the heads of myosin. So they're shaped sort of like this. They got the double head, and then you got a little bit of an angle to them. Okay, so that's a myosin protein, and it has these globular heads that form what are called cross bridges. They are the cross bridges. So here we can see this cross bridge connection portion of a myofilament, of a myosin myofilament. So they're the thick filaments, and then there are the thin filaments. Thin filaments are composed of a protein, lots and lots of these molecules of the protein actin. So each one of these little kind of small balls is an actin molecule, and they're bound together to form the thin filaments. And each of the actin molecules has a little binding site on it that is very attractive to the globular heads of the myosin. So actin and myosin, when allowed to, will bind to each other. And so it's this arrangement of the myofilaments, of the actin and myosin filaments, that creates the appearance of striatedness in muscle fibers. So this is a key statement here. A muscle fiber, so a muscle cell, will shorten as the thick and thin filaments slide past each other. So it's the binding and then the movement of the thick and thin filaments past each other that causes a muscle to contract. And I know we haven't got that explained yet. I don't expect you to completely understand that yet. But I want you to get the sense that there's nothing that there's, that the thick and the thin filaments, they don't get shorter. They only slide past each other, causing the muscle cell to shorten. Think of it like an extension ladder. An extension ladder is one of those tall ladders that has two elements to it, and you can lengthen it by pushing one of the ladder parts past the other ladder part, or you can shorten it by allowing the two ladder parts to slide past each other until they get to the shortest possible length of the ladder. So that is a critical piece to understanding how muscle fibers work, is that it's the myofilaments, the thick and the thin filaments, that slide past each other, causing the entire muscle to become shorter. The myofilaments do not shorten themselves. They always stay the same length. The shortening is this sliding past each other. Okay, so when we look at the construction of a myofilament, what we see is that they are built of these repeated units that are referred to as sarcomeres. So sarcomere is this repeated unit of this thick and thin filament construction. And one sarcomere runs from one Z line to the next Z line. So these little zigzag things are called Z lines. And so this is one sarcomere, and here's the second sarcomere. And it's the sarcomeres that become shorter. They're the contractile unit of a muscle fiber, and kind of the functional unit as well. And so understanding both the construction and then the physiology of sarcomere shortening is going to be important for this chapter. We're going to get there eventually, but we're not there yet to discuss about how shortening works. So sarcoplasmic reticulum, that is the endoplasmic reticulum, as you might have guessed, of a muscle cell, but it's called sarcoplasmic rather than endoplasmic. And it's this very elaborate form of smooth ER. So it's just smooth ER. And notice that it surrounds the entire myofibril, or myofilament, excuse me. The myofilament is composed of myofibrils, so we can see the thick filament here, and here's a thin filament right there. So those are myofibrils. The whole thing here, this construction, is a myofibril. So these are the myofilaments. This is the myofibril. I'm not sure. I might have gotten that messed up a second ago. So let's try that just to be sure. The thin filament is a myofibril. Is a myofilament. The thick filament is a myofilament. This entire construction is a myofibril. And the sarcoplasmic reticulum forms kind of a sleeve that surrounds each of the myofibrils in the cell. And its job is to take calcium ions that are found in the sarcoplasm, and it moves it from the sarcoplasm and into the interiors of this sarcoplasmic reticulum. So it's busy clearing the sarcoplasm of any calcium that it can run across. And there are these, along the length of the sarcoplasmic reticulum, there are these kind of holding stations that are referred to as the cisternae. Cisternae. That's an I, by the way. And you'll hear me talk about them as terminal cisternae sometimes as well. So these cisternae are enlarged regions of the SR that, as these parts here draw up calcium out of the sarcoplasm, then they concentrate that sarcoplasm here in the cisternae. So the cisternae are found at sort of the ends of these little sections of sarcoplasmic reticulum. And everywhere that a T-tubule, so here's a T-tubule here, everywhere that a T-tubule wraps around a myofibril, then it's going to be flanked on either side by terminal cisternae, by the cisterna. So here's a T-tubule, and then to each side of that T-tubule is the sarcoplasmic reticulum, the cisterna of the sarcoplasmic reticulum. And note the root here, cistern. If any of you have ever lived in a house that has a little underground storage for water, that would be a cistern. So back in the olden days, people would collect water off their roof. They would let it flow down into a cistern, usually buried near the house or underneath the house, and then they could pump water back up out of the cistern and use it inside the house. So it's another means of having indoor running plumbing. But so it's the same idea here. We've got a big old holding tank that gathers up and holds on to calcium. And so we'll see, as part of telling a muscle to contract, we're going to see that the cisterna on either side respond as the signal to contract comes down the T-tubule. So we're building up this muscle cell, and it's important that you kind of have a sense of what everything is doing and where everything is, so that you can understand the physiology of muscle contraction. Okay, so we're going to wrap up this lecture just now by looking at some of the stripiness that's caused by the thick and thin myofilaments that are in a myofibril. So here we again have a myofibril. It's pulled out. It's surrounded by sarcoplasmic reticulum, but notice they've cut it away. Here is a terminal cisterna. Here is a T-tubule. You can just make out on the other side there that it has another flanking cisterna on the other side of the T-tubule. So when we look at a sarcomere, we can see that it is arranged in a very particular way, where we have, again, the Z line. That's a plate of protein that is the attachment point for all the thin filaments. So thin filament and the Z line here. And then you can see that there's kind of these attachment points where the thick filaments are attached at the very center of the sarcomere. So the I band is this region that surrounds the Z line, and it's a region where there's nothing but the thin filaments. And there's also this protein titin, and it attaches the thick filament to the Z line. So here's the thick filament. It's attached back to the Z line as well, and it's attached by this protein fiber built of titin. Okay, so the Z line, it surrounds the I band surrounds the Z line here, and it's a region that is lighter in color because all that we see in this I band when the muscle is relaxed is thin filaments, and so it creates a lighter color in that stripy pattern. That's the lightness in between the dark stripes. The A band is the dark stripe. This is the region where the thick filaments are found. Sorry, don't know why that happened. So from one end of the thick filament to the other, that comprises the A band. When a muscle fiber is relaxed, the middle of the A band has what's called the H zone. That's this region right here. So the A band is anywhere that there is a thick filament. But notice that for part of that A band, we have overlapping thick and thin filaments. That makes this super dark region here that is the outermost portion of an A band. And then there's a slightly lighter region in here that is composed only of thick filaments. And so it's not as dark as these outer regions. It's a little bit lighter, so it's referred to as the H zone. So this is the H zone here. It's that region where only the thick filaments are found. And then right in the center of the sarcomere is the M line. So it's a dark line that's right there at the center of the A band, and it's composed of proteins that hold and anchor the thick filament. So just to review, we have I bands with the Z line right down the middle of the I band, and A bands with an H zone right in the center, and an M line at the very, very center. Okay, so we've seen the construction of a muscle cell. We've seen how they're built of sarcomeres, and how the sarcomeres are composed of these regularly organized thick and thin myofilaments that are arranged in a way that create the stripy pattern, where the lightest of the bands are provided by the I bands, where we only see thin filaments with a Z line right in the middle, that zigzaggy plate of protein that provides an attachment point for the thin filaments, and an attachment point via titin for the thick filaments. And then there's the A band with its H zone, lighter colored, where only thick filaments, and here are the darker flanking thick and thin filament areas. And then the M line that runs right down the center again. It's some connecting points that reinforce and support the thick filaments. All right, so I know this is a complex chapter. Hang in there. we're going to make some sense of it. When you come to lab on Wednesday, we're going to watch some videos that are going to also help to make good sense of this.